Hello, this is Daniel, and in today's video we are going to discuss about action potential and synapse. So by now we know how a neuron looks like and the several parts of neurons. So we are going to discuss how the neurons communicate with each other. So as discussed earlier, we now know how a neuron looks like. Let's do a brief review about the parts. The cell body, which generates the chemical neurotransmitters that are used for neural communications. And the cell body also contains the cytoplasm and the nucleus of neuron. And the dendrites, which are like the tree branches, which receives the chemical neurotransmitters from the previous neuron to receive signals, either that excites the current neuron or inhibits the current neuron from sending the signals to subsequent other neurons. And the third part is this extended axon, which is the part where the electrical signals that the neurons use to communicate with each other are transported along the end to the axonal terminal. Let's begin with a situation where the previous neuron sends a chemical neurotransmitter signal to this current neuron. That situation can be modeled like this. And yeah, this model has a lot of features, but we just need to focus on some of the features for now. These colorful circle objects are called neurotransmitters, which are chemical signals that neurons use to either excite or inhibit the following neuron to send or inhibit the sending signal part. When these neurotransmitters are released to the current neuron from the previous neuron, the current neuron has neurotransmitter receptors, such as this one, this one, and this one, and by receiving these neurotransmitters, subsequently ion channels that allow the influx of either positive ion or negative ion in our body could occur. So initially, without any changes or influx of ions, the membrane potential, meaning the inside of our neurons, are generally negative 70 millivolts, has the negative 70 millivolts of voltage. Now, due to these neurotransmitters binding to the receptors and activating certain types of ion channels, these membrane potentials could depolarize, meaning the membrane potential becomes less negative, or it could be hyperpolarized, meaning the membrane potential could become more negative. So if it is depolarized, if the neuron is depolarized enough and it crosses the threshold of the voltage threshold, then the ion channels opens automatically, triggering an electrical impulse called action potential, which travels along the axon and then reaches the axon terminal and begins another chemical signal transmission. Now the junctions that the signals are passed from one neuron to the other, like this one, is called a synapse. In most neural circuits, a synapse includes the end of an axon, which is this part, and the dendrite of an adjacent neuron, the postsynaptic neuron, which is this one, and a space between two called synaptic cleft, which is this space. So as mentioned before, chemical signals called neurotransmitters cross the synapse, which is called this process is called neurotransmission. So, giving more details into what I have discussed earlier, this part, axon hillock, is the initial point of action potential. Here, axon hillock has especially more number of those chemical ion channels that could lead to immense influx of positive ions at once, which increases the membrane potential to a very high degree, as shown by this graph. So this part is the initial membrane potential, about negative 70 millivolts, and sodium channels could open in dendrites due to the neurotransmitters, excitatory neurotransmitters. It goes up and up and up and up, and then when it crosses the threshold at axon hillock, the positive ion channels opens, increasing the membrane potential more, which depolarizes the neuron, and then, boom, this part. The electricity, the action potential is triggered, and then the action potential moves along the axon of the cell, of the neuron. Eventually, the neuron regains its potential to about negative 70 millivolts, but it requires time too. So this is the model of showing how the electricity action potential moves along the axons. So these are two types of different neurons, unmyelinated, and this is myelinated. It means that myelinated means that the axon of a neuron is wrapped up in this myelin sheaths, which acts like an insulator. Not an insulator, but it prevents the electricity to move directly in a direct manner from here to the end. It makes the electric signals jump over some of the spaces like this, and therefore the myelinated axons transport the informations faster than unmyelinated ones. So when the electrical signals reach the end of the neuron through this pathway, in other words, when an action potential arrives at the axon terminal, the voltage change in the axon terminal triggers ion channels in the membrane to open, which lets the calcium ions flow into the cell, like this. When the calcium ions binds to the packages of neurotransmitter molecules called synaptic vesicles, these ones, the vesicle fuses with the cell membrane. So this vesicle fuses with the cell membrane and then empties their contents into the synaptic cleft, like this. Then the pieces of axon terminal membrane cycle back into the soma like as new vesicles, which are refilled with neurotransmitter molecules. It fuses with the membrane, emptying their vesicle and then releasing the neurotransmitters to the next dendrites, and then it cycles back into the presynaptic neuron as new vesicles that could be refilled with neurotransmitters later. Now there are two types of chemical receptors, chemical neurotransmitter receptors. In an ionotropic receptor, a neurotransmitter binds directly to a part of an ion channel. So the channel is normally closed, but the receptor protein changes its shape when the neurotransmitter attaches, widening the tunnel so that the ions can move through. So ionotropic receptors are faster in letting the ions to flow into the neuron to depolarize the membrane potential. On the other hand, metabotropic receptors are more complex. The receptor and ion channels are different proteins located at a distance, but they are linked by a cascade of biochemical steps that are triggered when a neurotransmitter binds to the receptor. It is less rapid because it creates a cascade of events after the neurotransmitter attaches to the receptor, and the result may be opening an ion channel some distance away or activating other intracellular molecules. Now, going back to this slide, what happens to the neurotransmitters after they detach from the receptors in the postsynaptic neurons? Because neurotransmitter molecules only bind to their receptors for a short period of time, once they detach, the ion channels return to their resting state and stop altering the charge across their membrane. The neurotransmitters are either broken down or reabsorbed by the axon terminal in a process called reuptake. Now let's move on and talk about the different types of neurotransmitters, two types, excitatory and inhibitory. So excitatory neurons make neurotransmitters that open ion channels that depolarize the dendrite's membrane. On the other hand, inhibitory neurons make neurotransmitters that hyperpolarize that. The brain's most common excitatory neurotransmitter is glutamate, and the most common inhibitory neurotransmitter is GABA, which is gamma aminobutyric acid. This is a brief explanation of how the neurotransmitters are created at the end reaches the axon terminal. Some of them are reused from reuptake, and some of them are produced from the cell body neuron's nucleus, and then transported to the axon terminal through Golgi apparatus and following the microtubules along the axon. Because we are discussing about the neural communication in this video, I wanted to introduce the AMPA and NMDA receptor relationship, which strengthens or weakens the memories and learning. Glutamate that we have already discussed binds to NMDA receptors. So AMPA and NMDA receptors are all the receptors of neurotransmitters that are in the postsynaptic neuron. So glutamate, the excitatory neurotransmitter, binds to NMDA receptors. Now, if the synapse is activated quite slowly, the NMDA receptors play little or no role. Because if the synapse proceeds slowly, that means the change in the membrane potential changes slowly, which does not contribute to pulling off the magnesium ion that is blocking the NMDA receptor. But when the synapses are activated by several pulses very quickly, the NMDA receptors immediately sense this excitement. This greater synaptic activity, stronger charge change, causes large depolarization in the postsynaptic neuron, and this dispels the magnesium from the NMDA ion channels by a process of electrical repulsion. NMDA receptors are then immediately able to partake in the synaptic communication. In terms of AMPA receptors, they change the strength of neural communications. To strengthen neural communications, the neurons could increase the AMPA receptors' numbers or increase the efficiency of AMPA receptors. Enabling AMPA receptors to work more efficiently, such as passing more current into the neuron upon activation, or creating more AMPA receptors to be inserted into the synapses, leads to a larger depolarization, which is called LTP. The opposite of this is called LTD, which is reduction of the efficiency of number of AMPA receptors. This could occur within a single dendritic spine, and thereby alter synaptic strength in a highly localized manner. By increasing the number of AMPA receptors or the efficiency of AMPA receptors, the postsynaptic neuron could experience a quicker change in the charge, which could lead to faster neural communications, which leads to people coming up with ideas faster or answering the questions faster or being more adept or skilled to activating certain skills, including violins, sports, instruments, and any other things. This is the end of this video. Thank you for watching this very long video. Action potentials and synapses are a very complex concept, so thank you for watching and see you in the next video. Bye!